thanks, Jody, for that very thorough introduction. I'm glad none of the stories from Conor came up.
Maybe later.
Yeah, but I have some on him also. So, as Jody mentioned, we were just in at UQ where we worked on these guys, and this may seem like a cute little crocodile, but they are incredibly aggressive and very, very impressive animals. And we looked at salt and water balance in these animals. You all know probably that they can tolerate salt water and fresh water. But one thing that's claimed in the literature is that they do not drink when they are in salt water. And from a salt and water balance perspective, I kind of refuse to believe that. But now, having worked on them for a couple of weeks, I think that may actually be true. But the jury's still out on that. But anyway, we had a great time down in Brisbane, and we're looking forward to a great time the next couple of weeks out at Lizard Islands. I don't have to remind this audience about this situation. This is from the IPCC report in 2007. We have increasing atmospheric CO2. It equilibrates with the ocean. pH drops. We know that. Some of the values to keep in mind here is current level is much above what it used to be. And for the purpose of this talk, it's also important to note that levels between 1,000 and 2,000 microatmospheres is relevant for predicted conditions over the next couple hundred years. And what's also worth noting, I think, is that levels exceeding actually 2,000 microatmospheres is commonly observed present day. The Baltic, for example, and along the west coast of the North American continent, we see these kind of levels of CO2 on a regular basis. So this is not just relevant for say, year 2300, but also for current day, at least certain times a year. And Jody alluded to a review that we just published. This has been known for a while, obviously, and what I've always believed is that there's been insufficient focus on fish when it comes to ocean acidification and the impact of this environmental stressor. And based in part on that, and then based in part on what seemed to be an emerging interest in fish, we decided to submit this review to American Journal of Physiology, and it was published late last year. And I encourage you to look it up. But basically, we'll be basing this presentation largely on this review. This is one of the figures from the review, and what it's showing here is the number of publications summed over time over the last decade or so, perhaps a little more, in different groups of animals. And one of the points here is that fish have actually been fairly common in terms of studying impacts of CO2 and physiology. But I would point out that many of these studies actually done at very, very high CO2 levels that are kind of used as diagnostic to kind of explore mechanisms of acid-base balance regulation, in particular in these animals. A couple of key events to note here is a paper in 2001 by Brad Seipel and Pat Walsh. It was published in Science. This was a paper that was published in response to the idea of depositing CO2 in the deep sea. This paper pointed out it was probably not a good idea, and I'm glad they did. 2003, the publication by Caldera and Wickett points out that the changes in ocean surface water pH is actually faster now than it's ever been. Again, a very, very important observation. And then 2007, we're all aware of that report, and that's sort of when things started picking up in the field of ocean acidification research on organismal biology. And then I want to point out a paper here in 2009, came out here actually from Monday's group. That's the first paper, in my opinion, that really shows major effects of what we call lower OA relevant CO2 levels. And you can kind of see that the fish field sort of picked up after that. Still, I want to point out that considering the ecological significance and economical significance of fish, they are understudied. And I guess, depending on which animal is your favorite to work on, you're going to claim that for any of these scenarios here. But I do believe fish understudied, and I'm going to try and make that point, or we're going to try and make that point over the next half an hour or so. So what is the reason for this lack of interest, apparent lack of interest in impacts of CO2 in fish? This is from a review a few years ago by Shimatsu, where he's showing this is an example of mortality during exposure to very, very high CO2 levels in a number of different fish species. You can see we go all the way up to 26,000 microatmospheres. So, well over what we're considering from an OA perspective. And what you can see here is that there's no modest mortality at these very, very high concentrations. So fish can handle this. However, mortality is a very crude endpoint when it comes to considering fitness and general well-being of these organisms. But another reason why animal or people have not really focused on this is the notion that's well established in the literature that fish have very strong acid-base balance regulators. This is some work we put out some time ago now on midshipman. It's a benthic temperate species, coastal. And what you're seeing here is the plasma, the blood plasma, the extracellular fluid pH over a range of CO2 concentrations. And again, this is 5% CO2. This is exceptionally high, 50,000 microatmospheres. Very, very high. You can see it doesn't phase them with respect to plasma pH. And that has for a long time been assumed to mean that these fish are just fine in high CO2. It's not allowed. However, what happens during exposure to high ambient CO2 is that the partial pressure of CO2, the amount of CO2 in the animal, increases dramatically. And I'll get into in a minute why that's the case and also why perhaps it's a big, big problem for these animals. And you have the control values down here. Now, in the face of this high CO2, the constant pH is only possible because the fish accumulate bicarbonate to offset or buffer the impact of the high partial pressure of CO2. So fish are remarkable at doing this. Now, in an acid-base balance disturbance like this, air breathers like us, for example, what we would do is we would actually control the partial pressure of CO2. We can do that by, say, hyperventilating, for example, blowing off CO2 and restoring pH in that way. Fish are unable to do that, and it has to do with breathing water rather than air. And this is sort of I'm taking you back now to early physiology, early biology, and I apologize to those who had it and remember it, and I think those who have not, it's time they see this. If we're comparing here air breathers on the left to water breathers on the right, air breathers have a tidal ventilation, air is going in and out of the lungs. It's actually a really inefficient respiratory system, but it's fine because oxygen tensions in air are very, very high. There's plenty of oxygen around. An inefficient system can supply the oxygen we need. The consequence of this inefficient system is that you accumulate CO2, that's the dark line here. So CO2 levels in bodily fluids and in tissue is relatively high, at least compared to water-breathing animals. The problem with water is that oxygen solubility is very, very low. A liter of water contains only about 3% of the oxygen you're going to find in a liter of air. And for that reason, animals are sort of forced to hyperventilate with respect to oxygen. CO2 solubility is very high, so what that means is that to accommodate oxygen uptake or accumulation and consumption in water-breathing animals, CO2 is basically depleted out of the system. So CO2 levels are exceptionally low in water breathers compared to air breathers. And the consequence here is that water breathers like fish, in contrast to us, cannot adjust pH by adjusting CO2 levels, at least not to a great extent, because these CO2 levels are very, very low. Okay? What it also means, and I think this is really important to realize, is that as ambient CO2 increases, of course, under these conditions, there's an outward gradient of CO2. That's how it's released from these animals. But as ambient CO2 increases, it actually at very low levels becomes a problem because the gradient of pCO2 would be reversed unless the animals adjust pCO2. That would not be the case for air breathers. So yes, fish are exceptional acid-base balance regulators. They can certainly survive high CO2 levels. They're very good at accumulating plasma bicarbonate, but when they do that, they also accumulate CO2, which may present a problem as we're going to discuss as we go forward here. Now, just going back real quick, we had this stubbornly defended pH despite very high CO2 levels, and these CO2 levels, of course, are not relevant for OA scenarios. But if we look at first how these animals can cope with these high CO2 levels and accumulate bicarbonate, you've got to consider the gill of these animals. So this is where the gas exchange is occurring, oxygen is taken up, of course, CO2 is released. But what's really important from an acid-base balance perspective is that the CO2 can also be hydrated within the gill epithelium, these epithelial cells here, to form bicarbonate and protons. That hydration reaction is mediated by carbonic anhydrase, which is an enzyme, and will basically lead to the possibility of excreting bicarbonate into the water from the gill cell and transporting protons from the gill cell into the blood in a situation of an alkalosis. So if these animals have a need to correct an alkalosis, they're going to drop the base and they're going to retain the acid. Conversely, if we talk about an acidosis, and this is what happens during high CO2 exposures, the animals can do the opposite. They can retain bicarbonate and they can eliminate protons. This is the basis of the very efficient acid-base regulation in water breathers, and most of them, and certainly in fish. Now, of course, at any given day, these animals are doing both to a combination, or in a combination that accommodates the physiological conditions and the environmental conditions. But this is the basis of, as I said, the very, very strong acid-base balance capabilities of water breathers, in particular fish. Now, looking at ocean acidification relevant CO2 levels, I just want to kind of stress a point here. If you're looking at PCO2 in the plasma, as these animals are exposed to increasing CO2 levels, and here we are dealing with environmentally relevant CO2 levels and certainly OA relevant CO2 levels, you see that the partial pressure internally is actually exceeding control values significantly at levels as low as 750 microatmospheres. Nevertheless, pH is defended, as we saw before, at the high CO2 concentrations, no surprise there. But what's interesting here is that this protection of pH, despite the increase in CO2, again, is achieved by accumulation of bicarbonate. And again, this bicarbonate accumulation is actually evident at 750 microatmospheres, yet they can survive 50,000 microatmospheres. But again, as I mentioned earlier, surviving is one thing, being happy and healthy is another thing. And I should say that these samples here are taken after 24 hours of exposure. The animals have been exposed for 24 hours. They have a time to adjust, and this is sort of the adjusted physiology you're looking at here. If you're looking at the time course, the bottom panel here shows the blood pH over 48-hour period in this beautiful animal, the Gulf toadfish here, exposed to 1900 microatmospheres. And what you can see immediately as PCO2 is increasing, there's an acidosis in the blood, which is what you would predict. And then there's a recovery here after just, say, 4 to 8 hours. That recovery, of course, is mediated by the accumulation of bicarbonate. So what's happening in the animal, sort of from a schematic perspective here, is as CO2 is increasing outside, it's increasing inside. There's no way they can work around that. That causes a drop in pH in the extracellular fluids. This phenomenon is referred to as a respiratory acidosis. The metabolic compensation for this is accumulation of bicarbonate. I've shown you the data already. And that actually returns the pH to normal pH levels. And this is why people think that these animals are going to be fine in high CO2. They don't die, they have normal plasma pH. But it's very important to realize that that scenario is associated with a second steady state, and that steady state is characterized by high bicarbonate in the extracellular fluids and by high partial pressures of CO2. That does not change. These animals are living under those conditions. Now, this is a figure from this review also that's kind of looking at the impact of CO2. You have the concentrations on the x-axis here, or the partial pressures for CO on the x-axis, and you have different categories of effects. We kind of grouped all the observations in the literature, or at least by far most of them, in categories as base balance, calcification, I'll get back to that, neurosensory and behavioral functions, growth development, survival, reproduction, metabolic rate, swim performance, mitochondrial function, cardiorespiratory impacts, and osmoregulation. And you can see that some of these fields certainly are in need of much more attention. But what I think is really interesting is, despite the fact that people perceive these animals as being very, very tolerant to high CO2, you see a lot of reports of effects down in climate change relevant CO2 concentrations or CO2 levels. As a base balance, which we actually believe is probably at the core of many other of the effects. We've looked at how that's disturbed already, and that it's also compensated. I will point out that this compensated state in itself is an effect, and you've got to keep that in mind. In my lab, we look at as base balance, calcification, and osmoregulation primarily. So I'm going to be focusing on that for the next 10 minutes or so. And you may ask the question, hold up, calcification, fish are not calcifiers. And in fact, that's why they have not received a lot of attention in the OA literature. All the calcifying organisms, as you know, have received more attention. But fish actually are calcifiers, and I'll explain that in a second. This is perhaps one of the more obvious ways in which fish are calcifiers. The otolith is a small calcium carbonate structure that's found in the inner ear, and it's used for balance and acceleration type measurements in animals, or in the fish at least. The otolith is formed within a saccular epithelium, and this is a cell model of the ionocytes, or the cells that transport carbonate and calcium into the lumen of this saccular epithelium for the formation of the otolith. And basically, the take-home message here is that bicarbonate is imported into this lumen where the otolith is formed, along with calcium, and that allows for the calcium carbonate precipitation in these structures. And remember here that fish exposed to low levels of CO2 and high levels of CO2, they have this second steady state of higher PCO2 and higher bicarbonate. Now, the higher PCO2 in the surrounding blood is going to lead to an increase in partial pressure in the cell, just because it's going to change diffusion gradients. Bicarbonate in the extracellular fluid here, as that's increasing, is increasing the substrate for these transporters that ultimately allow for greater accumulation of bicarbonate in the fluids, in the saccular epithelium, and thereby for greater formation of otoliths, or otolith formation at a higher rate. And there are a number of studies showing that otoliths are increasing the size as ambient partial pressure of CO2 increases. Unfortunately, or fortunately, I guess it depends on the perspective, at this point there are no reports of this actually meaning anything in terms of the performance of the animal, but I think it's pretty intuitive that there's going to be some sort of impact of changes in a structure that's important for a sensory system of this magnitude. The other way in which fish are calcifiers and contributes actually to the inorganic oceanic carbon cycle is via osmoregulation. So I'm just going to take 2 steps back and introduce that. Marine fish have the same internal osmotic pressure as we do, about 300 milliosmoles, comprised mainly of sodium and chloride. They're surrounded by salt water, seawater that's a lot more concentrated. So they lose fluid by diffusion all the time, and they also gain salt. The compensation for this fluid loss is drinking. They drink seawater, and they absorb sodium and chloride in the intestine to drive water absorption. It's called solute-coupled or salt-coupled water absorption. This has been known for over a century. If you look at the seawater as it's passing through the intestine, and this is a very busy diagram, we're not going to focus on all of it, what you can see here is that sodium and chloride is depleted as fluid is moving through the intestine. That's to drive the water absorption. What you can see is that calcium, which is about 10 millimolar in seawater, is actually a lot lower than it would be if it was just seawater found in the intestine. And this is actually not due to uptake of calcium. I'll get back to that in a second. And perhaps most remarkably, what you see is bicarbonate, which is only a few millimolar in seawater, is accumulating up to 80 or in some cases over 100 millimolar in the intestinal lumen. Okay. Now this very high concentration of bicarbonate, along with the calcium that's ingested with seawater, and the fact that calcium is actually left behind in the intestinal lumen as water is being absorbed, leads to precipitation in the intestinal lumen of calcium carbonate. So these fish form calcium carbonate in the intestinal lumen, and it is released to the environment. This is the calcium carbonate formed by a toadfish over a 48-hour period in this tank in seawater. This is normal salinity seawater. So this is substantial. And if you don't believe me, this animal is producing the calcium carbonates in the intestinal lumen. You see the calcium carbonates right in this area right here, and they're released with the rectal fluids through the rectum to the environment. Now, this is a benthic toadfish, and it's an adult. This is occurring also in larval pelagic fish. This is a cobia larvae. This is about 12 millimeters long. This is a CT microscan that's showing right here a calcium carbonate structure. And I'm just going to play a video here just to convince you that this is actually calcium carbonate that's contained within the intestine. So this is a simulation, but what you can see is that as this animal is rotating, this is not in front or behind the animal. This is in the viscera. So now it's below here, and there we go. And we're going to kind of zoom in and look from the front of the animal, and you can actually see that this is sort of coiled up and following the lumen of the intestine of this larval cobia. So these animals produce calcium carbonate in the intestine, and it's released to the environment. That's the bottom line. We've looked at more than 40 species now. They all do it. Some years ago, well, actually, let me explain how they do this first and how significant it is. This is a very simplified diagram of the cell in the intestine here. The cell is producing CO2. It's a metabolic waste product. That CO2 is hydrated within the cell to form bicarbonate. That bicarbonate is released into the intestinal lumen. In the intestinal lumen, it's combining with the calcium that's contained in the seawater that they're drinking. They have to. And it forms the calcium carbonate. Now, the significance from a osmoregulatory and fish perspective here is that what you're essentially doing is you're taking CO2, which is a gas that's inert from an osmotic perspective. It does not exert an osmotic pressure. You're exchanging that for chloride, which is an osmolyte. That osmolyte will attract water and allow for water absorption into the intestinal cell and ultimately the fish. So this is sort of a net gain of an osmolyte. This helps water absorption. Remember, these animals are water stressed. In the intestinal lumen, what you get is you get a precipitation, so you're basically taking calcium and bicarbonate out of solution. They no longer exert osmotic pressure. Instead, they form this precipitate that sinks out. The advantage or the gain for the animal is a reduction in osmotic pressure by about 100 milliosmoles. Without that, these animals, and we've shown this experimentally, without this reduction in osmotic pressure, that's the product of this precipitation, these animals could not osmolate in salt water. There would be no marine teleost fish in marine environments. And to just illustrate how powerful this is, the European flounder is a really good model. They're very, very urinating. not only can they tolerate freshwater and saltwater, but they can tolerate acute transfer from one media to the other. So what we did in a study we published a few years ago in Science is we took a European flounder and we placed it on an X-ray instrument. The European flounder is great not just because they're European, but also because the viscera here is not covered by bones at all. You can see the viscera. It's completely X-ray opaque. And you can see here that there's nothing going on here. After this image, we transferred this particular animal to full-strength seawater for 3 hours. We left it for 3 hours to dehydrate, drink, and deal with that physiological challenge. And then we transferred it back onto the X-ray instrument here. And what you can see here is these X-ray opaque structures here. That is the calcium carbonate that this animal is forming to osmoregulate. And this is, perhaps the most significant contribution when it comes to calcium carbonate formation by fish. All this just to say that fish are calcifiers. There's no way around that. Okay. Now, this calcium is actually released to the environment, and we've estimated the contribution of fish to the inorganic calcium carbonate or carbon cycle in the oceans. And it basically amounts to up to about 15% of the total production of calcium carbonate in our oceans. So this is not insignificant. And of course, one of the questions now is, what is the impact of elevated ambient CO2 going to be? One, on the production of these carbonates by fish, and two, the fate of these carbonates. Are they going to actually reach the sediments? Is this going to be a sink for CO2? Or are they going to dissolve at depth as these carbonates sink out to lower saturation levels at depth? To sort of frame this discussion, this is a diagram of what we know about the cellular transport processes in the intestine of marine fish, and this is very basic. But don't panic, most of this is not really relevant. Again, what's relevant here is that as fish are exposed to CO2, they cope, they compensate, but when they do that, they have elevated plasma bicarbonate and elevated plasma partial pressure of CO2. Partial pressure of CO2 in the plasma is going to back up, so you're going to get an increase in CO2 in the cell. That CO2 is going to be hydrated to form bicarbonate that's now available for excretion into the intestinal lumen. The same is true for bicarbonate in the blood. It's going to enter the cell through a sodium bicarbonate co-transporter on the blood side of the tissue. It's going to raise the cellular concentration of bicarbonate that again is going to facilitate bicarbonate excretion into the intestinal lumen. And remember that this bicarbonate is what's combining with calcium to form calcium carbonate, and thus the release of calcium carbonate and a contribution to the inorganic oceanic carbon cycle. Now, whether ocean acidification is actually affecting this process or not has been one of the key areas of focus for Rachel during her PhD. So she's really much better at this than I am. So I'm going to step down and let Rachel take over.
Thanks, Martin. Thanks, Joey, for the introduction. So like Martin said, the majority of my PhD has been looking at how this new steady state that Martin has been talking about in the blood plasma may affect intestinal-based secretion and calcium carbonate formation in marine fish. And my model organism is also this lovely Gulf toadfish here. And one way that I was able to look at this particular question in an earlier study, my PhD, was to take a look at this toadfish here. You see as a rectal, what I call a rectal collection sac attached. It's a surgical procedure where I'm able to look at the total amount of both fluid and calcium carbonate produced by this fish over a set period of time. So for this study, I exposed fish to either control or 1900 microatmospheres for a 3-day period and collected the contents, which you see kind of an example in this particular tube. And one thing that we see first, taking a look at this graph with the rectal base excretion rate, or essentially the rate of bicarbonate secretion, is that we don't really see a significant difference in the solid portion. So Martin mentioned that could be something important for the global oceanic carbon cycle. In this particular species, we don't see it, but we don't know if that's the case in other species. And there are other labs taking a look at that now. One thing I do want to point out, though, is we do see a significant increase in the fluid component of this particular excretion. So we see an increase in bicarbonate secretion, either from transepithelial movement or possibly from the increased hydration of CO2 from this enzyme here, chloronic anhydrase, which could account for the increase that we see in this particular graph. And the main point here is that, again, this new steady state in the plasma is basically leading to base loss in the intestine. And as Martin said, during CO2 exposure, fish are trying to retain elevated levels of bicarbonate in their plasma in order to maintain that pH. So to me, when I first saw this result, the question that kind of came up of why are fish losing base via the intestine? Because this seems kind of counterproductive on a whole body basis. So what I ended up doing after this was looking at toadfish exposed for a longer period of time, so chronically exposed toadfish to either control or 1900, again using the same experimental level, exposing them for a couple of weeks, and then taking a look at their tissue, so doing an isolated tissue setup to see how the tissue responds when it's in this pH stat titration system setup. And really, this is a way of measuring the intestinal-based secretion rate in vitro rather than in vivo. And I predicted originally that because this base loss is likely counterproductive, fish would somehow downregulate or reduce the response when exposed to CO2 under long-term conditions to try to retain that base in the body. And as you can see, when we take a look at the graph here, under CO2 conditions, tissue from fish exposed to 1900, that bar is not lower than control, it's actually increased, which indicates some sort of stimulation. So there doesn't appear to be any regulation to reduce base loss through the intestine. And my next question, follow-up question to this is essentially, could there be a metabolic cost associated with that? And this is research that's still going on, that's still ongoing. I'm kind of in the very last portions of this since I'm finishing up my PhD. But this is basically another way to measure, basically take a look at this issue in an in vitro or isolated tissue setup. I again exposed these fish for 2 weeks to either control or 1900 conditions, and then put the tissue in an isolated epithelial respirometer to just basically measure oxygen consumption rates. And this is some preliminary data just showing a possible trend. I still am working up the rest of this data, but it looks like there may be some sort of response here. So I'm now going to switch gears a little bit. We've covered a few of the categories. We obviously can't do all of them today because I think we'd be here for quite a while. I'm now going to move into neurosensory and behavior because this is one of the more robust areas of research, and I know most of the studies probably from this area have come out of from this particular school or collaborations from within this school. So in the 30-plus studies, I apologize in advance if I can't touch on all of them because I think we would be here for quite a while. So I'm going to use a few representative examples to review and then talk about a neuronal cell model that Martin and I worked on in the review paper. So to briefly review a lot of the studies that have been done here, we know that we've seen impacts across multiple sensory systems in fish. This is an example from one of Phil's earlier papers looking at how basically you start seeing at higher CO2 levels fish unable to properly respond to a predator cue. And then on the right graph here, we see that this lab demonstrated basically sensory abnormality does translate to increased mortality in a field setting, so it is environmentally relevant. In addition to olfactory disturbances, there's also been demonstrations of auditory disturbances and also visual disturbances, so again, multiple sensory systems. In addition to looking at sensory systems in isolation, there's been other measurements of broader cognitive functions, so for example, lateralization, learning, and other behavioral examples such as boldness, activity, and anxiety. And overall, what this and what many of the papers Phil has been involved with basically concludes is that there is a lot of evidence that ocean acidification is somehow affecting central neuronal processing in fish. And in one of his reviews, Phil, and in a lot of his papers, he's gone through a lot of these potential impacts, so impacts to dispersal, recruitment, social interactions, predator-prey dynamics, habitat preference, and settlement timing. And because there's such a robust response here, because the impacts are so important, it leads to the question of what is the underlying mechanism? Why is this happening, and why do we see this response? And this was an important paper put out to hypothesize one of the major, provide a hypothesis for these neurological effects by Göran Nilsson in 2012. And he basically hypothesized that changes to ion gradients due to the CO2 compensation, so what Martin already talked about earlier, can affect membrane potentials, disrupt neuronal receptors, and lead to alterations in behavior. And he had an interesting idea that animals with the best developed acid-based regulation could be at most at risk, basically. So basically what his hypothesis was creating a link between the ion gradient changes that we see during CO2 exposure and the function of the GABA-A receptor. And this is important because GABA and its associated GABA-A receptor are the primary and is responsible for the largely inhibitory neuronal responses throughout the body. So I'm just briefly going to review this study to link because it's going to link to our neuronal cell model. Under control conditions, looking at both, I believe, olfaction and lateralization here, when the GABA neurotransmitter binds to the GABA receptor, under control conditions, when this channel is open, negative ions are moving into the cell, hyperpolarizing the cell, and creating what is a normal condition or an inhibitory response, and what we would consider, let's say, normal behavior. Under high CO2 conditions, the hypothesis is when GABA binds and this channel is open, because of this compensated state, rather than these ions moving into the cell, they are moving out, thus depolarizing the cell and creating an excitatory response and an abnormal behavioral phenotype, so unable to properly undergo proper lateralization or understand olfactory cues. And the third important thing that happened in this study was the use of the antagonist here. This is the GABA antagonist, gabazine. It effectively closes the GABA receptor. And by doing this in combination with exposure to CO2, it prevents these negative ions from leaving this channel. And what he was able to see is an alleviation of the excitatory abnormal behavioral response, alleviated and back to basically what would be considered the normal phenotype. And this has since been demonstrated in other studies, or at least the involvement of GABA-A receptors has been demonstrated in a couple of other studies in other species. This basically led Martin and I to really think about taking it one step further and looking at some more underlying neurological possible mechanisms on what's going on here at the cellular level. So we basically created a cellular model. So some of the major goals and kind of things we were thinking about when we were working on this is, first, can we use calculated and measured physiological values for fish exposed to CO2 to support results from these behavioral assays? And we have these values from the toadfish, as Martin described earlier. Second is to think about what other factors may be at play when we look at the cellular machinery we presume to be associated with response. And three, if we understand some of these factors, can we use this as a model, as a baseline for further study, and possibly as a predictive tool? So in order to build this model, there's a few things you need, and I consider it kind of pieces of a toolkit. The first thing you need is knowledge of how to calculate the equilibrium or the reversible potential for E-GABA, which we are able to do. The second is to find a neuronal cell model. Third is to, again, have extra and intracellular values of relevant ions in fish during control and CO2 exposures. And finally, to look at permeability ratios for the GABA-A receptor, since this is the one we believe is impacted. So I'm going to kind of take this step by step, because the cellular diagram gets quite complicated here. But first, just taking a look at the equilibrium potential equation for GABA. This is modified from Front and Kayla 2007. There's a couple things I want to point out about this formula. First, the R and the F are just constants. This equilibrium potential is dependent on temperature. But what's most important to take away from this is that both the concentration inside and outside of the cell of chloride and bicarbonate are important for setting this equilibrium potential for this receptor. Something to keep in mind. So now that we've taken, and I also want to add that these are the permeability ratios for those ions. The second thing we need is a neuronal cell model, again from the same paper. And as I don't want to get into the details of what all of these transporters are doing, but just to make the point that we know that chloride and bicarbonate are important, and we clearly see in many of these transporters opportunities for these ions to move in and out of the cell. So using this equation in combination with a cellular model essentially allows us to predict the direction of the ions under a certain given set of environmental conditions. And that's what we attempted to do under control in CO2 conditions. So kind of moving into the next step of the model, using our calculated values, Martin talked about the study that we did earlier on the toadfish, and again we used this representative CO2 level. What we see here, if we first just look at the extracellular, outside of the cell, we see that there's an increase in bicarbonate, an increase in PCO2 as we expect in that new steady state. And also, as Martin mentioned earlier in the talk, there's a decrease in chloride. Now, if we move into the inside of the cell, we see a very similar trend. We see an increase in bicarbonate inside the cell with CO2 exposure, an increase in PCO2, and a decrease in chloride. So now what we're able to do, we have all the tools that we need to plug all of these values essentially into our E-GABA equation and see what our equilibrium potential is for the GABA receptor. And once we do this, again, all of these values are still the same as the previous slide, is if we take a look at the control situation on the left side, and we're considering -70 our normal resting potential of the cell, we see that under control conditions, looking at two theoretical and physiologically relevant permeability ratios, so considering two different cases, that we see a negative deviation from the resting potential in control conditions. And this is, in terms of GABA, this would be a hyperpolarizing effect. And again, if you remember from earlier, that is associated typically with normal behavior in the assays that have been done thus far. Under the 1900 microatmospheres condition, plugging in all of our values into the equation, what we see here is a positive deviation from the resting potential. And this is considered a depolarizing or an excitatory response, which is again potentially associated with the behavioral abnormalities. So the take-home message, I think, from all of this is that even at very small, what is seemingly small increases in these values can could potentially drastically change neuronal function in the body. And in addition to thinking about gradients and transporters, there's a number of other factors that could be at play. We really only looked at the equilibrium potential for GABA and just a general cell diagram, but many other avenues possibly for future research is we know that there's some between species and also within species variation in terms of sensory system responses that it may be useful to look at some of these other factors, including dynamic regulation of these transporters, so any of the changes here or here. Possibly variation in permeability ratios for the GABA receptor itself. Circulating GABA, the actual neurotransmitter that binds, taking a look at circulating levels of that. GABA-A receptor expression levels in different tissues or areas of the brain, for example. Looking at GABA synthesizing enzyme and intracellular GABA transporters, and finally looking at variation in GABA-A subunit composition, and there's quite a bit of mammalian literature that this is important for factors like age, sex, brain development, and by region. And it might also be important for future work on pharmaceutical targeting. And that brings me to over here, and Martin's going to take that forward for that. Sorry.
Yes, so I think that sets the stage for an explanation of why we came here and why Phil and I agreed to work together here. We're basically here to test this prediction that this change we see in bicarbonate and the associated change in chloride that occurs during CO2 exposure for pH compensation, whether these altered gradients of bicarbonate and chloride form the basis of altered behavior in these animals during CO2 exposure. And there's a lot of information suggesting that is indeed the case. But as you know, it's very hard to get a solid proof for anything, and that's also going to be the case after we're done. But I think we might have a chance to take one step closer here. So the idea is to characterize the behavioral response to CO2, and then over a range of CO2 concentrations with differences in the magnitude of the response, and then to measure the brain bicarbonate concentrations, and that involves measuring brain pH inside the cells, to basically kind of look at how that may or may not correlate with behavioral phenotypes. And we are going to be taking samples also to assess any compensatory or regulatory responses by the central nervous system. So we're very excited about this. And then just to wrap up here, fish do compensate very effectively for any acid-base balance disturbance, including high CO2. But that doesn't mean they're not sensitive to CO2 under climate change scenarios. They are. And that's because the compensatory response, the new state, basically involves a change in bicarbonate and chloride concentrations both inside and outside the cell. And we believe that's actually probably responsible for a lot of other effects, including behavioral disturbances. I'm not going to go into the acid-base balance. We've talked about that. Calcification, fish do calcify. They calcify otoliths, and the calcification rate of otoliths is increasing with increasing CO2 because of this acid-base balance compensatory response. They produce calcium carbonate that's important for the inorganic oceanic carbon cycle, and that's possibly sensitive to elevated CO2. It doesn't appear to be in toadfish, and we have some ideas about why that is, but that's not to say that it's not important in, say, tropical or subtropical species. We know that osmoregulation is impacted. In fact, the lowest partial pressure of CO2 we've looked at shows impacts on osmoregulation. So we need to go lower to see just how sensitive this important physiological function is to CO2. And then Rachel's just discussed what we believe is possibly a link between acid-base balance compensation and disturbance of behavior and cognitive function in fish during CO2 exposure. I think with that, we'll welcome discussion and questions.